Hello and welcome to the live 2021 Nephrology Campus on current trends in nephrology. My name is Christoph Wanner. I'm chairing today and the entire program together with Professor Peter Stenwinkel from the Karolinska Institutet in Stockholm, Sweden. Today we discuss webinar number 17. So we are far advanced in the series, and the series of all webinars are online. And if you register to the site and the program, you can listen to all the topics we have been discussed over the past month. So you as the audience, you are automatically muted and not visible. But you can ask questions during the discussions and during now the talks. And you have a button on the right bottom corner, and in the chat room you can bring us the questions you have here to the studio, to me, and we can discuss it later on. Today we have two prominent guests, guest speakers from the Renal Research Institute, New York. And this is Professor Nadja Grobe, or Grobe. She is a scientist of the Renal Research Institute, and the director is with us, Professor Peter Kotanko. They will both discuss precision medicine topics today in this webinar number 17. So we will have both talks initially pre-recorded, and we can listen to the two talks. And Professor Grobe gave the opportunity to a senior researcher, Xiaoling Wang, and she gave the opportunity to come into the question and answer session we have at the end of the two talks. And it's my pleasure then to discuss with Peter Kotanko and Xiaoling Wang the questions you will have and you will give us in the chat room. So I think without any ado, we go to the two topics, and thank you for being with us today.
Thank you so much for this kind invitation to talk about precision medicine at the Renal Research Institute. It's a great pleasure to introduce to you Nadja Grobe. She is a PhD and supervisor of the laboratory research here at RI. My name is Peter Kotanko. I'm a physician and research director at the Renal Research Institute in New York. So these are our disclosures. And from there, let's move on to something that we think is really important. In the 21st century, there is a convergence of various trends. And these trends are precision medicine, pervasive sensing, data recording, and artificial intelligence. And the way how we conceive precision medicine is actually to be part of this much broader movements that eventually will bring us the convergence of these areas, such as precision medicine, pervasive sensing, mathematical patient twins, or mathematical modeling on a personalized level, electronic health record, and so on and so forth. And all this integration is made possible through multimodal data networks, AI, mathematics, and enabled through massive parallel cloud-based computing. Now, over the next few minutes, Nadja will speak a bit about the biochemical aspects of precision medicine, and then I will follow to talk a bit about some more device-based aspects. So, Nadja, the floor is yours.
Thank you, Peter, for the kind introduction, and thanks to the organizer for the invitation. In oncology, personalized treatments have been explored because it was clear that not all patients actually react in a similar fashion to the one pill fits all approach. And therefore, there is a need to really identify the specific cause that results in a different response to medication or therapy. Physicians don't treat populations, physicians treat individual patients, and it's not clear where the patients actually lay considering the population. The future of medical treatments will be oriented towards shifting the focus really to personalized medicine by targeting a phenotype that goes beyond the usual indicators and considers genetic makeup and even other omics inputs. Here at the RRI, we actually use these advantages of an omics approach. And on the left, you see something that we usually throw down the drain. It's a PD effluent. It's actually a very rich material, a biobank, if you want to say that, contains different materials such as cells and a dissolved fraction. And with this material comes a lot of opportunity shown on the right. Different technologies can be used to analyze the RNA, proteins, small molecules, and really generate these phenotype-associated molecular signatures shown down below called PAMs. In an omics approach, it's actually a really interesting discovery tool. As I mentioned earlier, the peritoneal dialysate fluid, spent fluid, is rich in biomolecules that come from each individual patient. And it's indicating peritoneal transport status, health, any ongoing pathological processes, and even underlying diseases and comorbidities, as opposed to the left, where you have a hypothesis-driven approach, where you have targets for inflammation, such as cytokines, infection, where you look at white blood cells, pathogens, and even membrane remodeling, where you have certain targets that you focus on. On the right, you actually only look at the clinical phenotype, for example, inflammation, and then you use an open omics approach to really create these PAMs, the phenotype-associated molecular signatures. And we take advantage at the RRI of this open omics approach. Here at the RRI, we recruit peritoneal dialysis patients and collect a small amount of their leftover peritoneal dialysate, their spent PD effluent, and then we go into either biobank the dialysate, or we submit it immediately to automated sample preparation using a liquid handler. Then we have an automated data acquisition system where we, in this case, use liquid chromatography mass spectrometry. And in a discovery mode, using advanced data analytics, we aim to discover and validate prognostic and predictive biomarkers. With these, we are really trying to look into dialysis monitoring and customized prescription to help with and improve patient outcomes for each individual peritoneal dialysis patient. This is on the next slide. I'm explaining what we actually attempt to do with our lab team, with Shaoling Wang, Xin Wang, Mia Gabacho, and Zahin Haq. They are working extensively on this project. We are using an untargeted metabolomics approach to develop these fingerprints or signatures that we correlate with patient outcomes. And how do we do this? So you see down here on the y-axis in abundance, and on the x-axis you see the acquisition time. So each color, each individual peak, we call them peak, represents metabolite. In an untargeted approach, we really don't care what the metabolite is at this point. We just try to discover certain abundances versus these signatures. And in an untargeted approach, we overlay these molecular signatures for each patient over a certain amount of time. So let's say we have, in this case, 2,000 metabolic features, and red means high abundance, and blue means low abundance, and anything in between means anything in between high and low abundance. We can do this for many different patients and get these different unique patterns or molecular fingerprints. And in A on the left, you see the 2,000 metabolic features that we identified using LCMS for 7 patients. And we really only need a small amount from a routine visit. So no extra visit is required for the patient or the care team. The spent dialysate undergoes our automated sample processing. In this case, we had 2 patients where we didn't know the transport status for these patients. We only knew that 5 of them were either fast or slow transporters. And then in B, using hierarchical cluster analysis, we delineated the differences of the molecular fingerprints with the peritoneal transport status. And I know this is not ideal because this unknown patient number 5 moved over to the upper half into the slow transporter phenotype, and this unknown patient number 2 moved in the lower half of this cluster analysis, illustrating that this patient looks more like a fast transporter type. And with this, we really want to follow patients over time to understand what their transport status is and help improve the prescription for these patients depending on their changes in transporter status. And we do this by using machine learning. This effort is spearheaded by RI's Senior Manager of Biostatistic and Applied Artificial Intelligence and Machine Learning, Dr. Hanji Zhang. And in this case, we look at the clinical outcome transport status, and we will approach this with supervised machine learning. And we'll start with a training data set to really evaluate these molecular signatures that correlate with transport status. We'll use classifiers to cross-validate and predict transport status as per PET. That's the traditional approach to measuring transport status, and then evaluate these classifiers using a testing data set. Currently, we are working with the group of Matthias Kuss, Data Solutions, and in particular Lena Scherer, who will help us with associating these individual molecular signatures with membrane transport status. And here you can see, for example, many different individual molecular fingerprints. And here you see the result of a classical PET test. And we'll use this to associate these fingerprints. At the RI, we also have lots of clinical trials. And with this clinical trial, we attempt something big, and we are very fortunate to not only work with Peter, but also with Dinesh Shatoth, who is the Associate Chief Medical Officer of FKC. That opens up unique opportunities for us to really recruit not only from RRI clinics, but also from FKC clinics. And of course, we have an amazing clinical research team at the RRI that really helps to roll out the study, consent patients, and get through all of these single steps. It's a complete workflow to really get the samples from the patient to the RRI lab that helps us to create these fingerprints. Our goal is very ambitious. We are trying to recruit from 100 RRI and FKC clinics. We are aiming for 1,000 patients and 3 to 4 years of repeated sampling and follow-up. And here you can see that we are trying to reach as many clinics as possible, and there's no geographical limit. So here we are in New York, where we target RI clinics, and then we expanded already to a couple FKC clinics. On the next slide, I told you that we also do, next to metabolomics, we also do proteomics analysis. In this approach has been spearheaded by our senior biomedical research scientist, Xiaoling Wang. In this case, we did a bench study using different dialysers and the 2008 hemodialysis machine. The dialysers we used were a Theranova 400, and we'll call this one from now on a medium cutoff MCO dialyser. And we also used the Fresenius Optiflux F180NR, and we will call this now high flux dialyser. And we chose those because of a similar surface area. And this is a bench test using plasma. On the next slide, you see the results, and I would like to focus your attention to A and lane 3 and 5. In these, you see that at certain time points of the dialysis, the dialysate contained proteins. This is an SDS page. You see protein markers, and here you see the plasma. So on lane 3 and lane 5, you see a lot of proteins that are around 62 molecular weight. And on lane 4 and 6, you see also small amounts of proteins, but these come from the high flux dialyzer. So clearly that SDS page shows that there are more proteins lost in the dialysate using a molecular cutoff dialyzer as compared to high flux dialyzer. And the setup of this experiment is shown here, but during these 240 minutes of experiment measuring total protein levels in dialysate, it's always shown that the MCO shown in orange contains more protein in the dialysate compared to the high flux dialyzer shown in blue. And this is also represented in this table here when we measure total protein. Now, Xiaoling did also something very smart. She used proteomics to identify dozens of unique proteins in the MCO dialysate. So remember, the MCO dialysate contained much more protein than the high flux dialysate. And we have two ways to analyze this protein using liquid chromatography mass spectrometry. In the first approach, we call this top-down approach, where we analyze intact full-length proteins. Xiaoling chose a bottom-up approach, where we do an in-solution digestion and then use electrospray ionization to really analyze these digested smaller peptides of a whole protein. And with that data analysis, we were able to identify very interesting proteins that are also not only harmful, but beneficial for the patient. So we think that this requires further investigation, because if a patient loses during a treatment not only harmful proteins, but also beneficial proteins, that would require a more thorough investigation. Our future plans are indeed to start a clinical evidence generation with prospective trials where we look at individual patients and different modalities and look at how different modalities affect dialysis efficacy for these different patients and eventually their clinical outcomes using advanced data analysis, and in this case, again, liquid chromatography mass spectrometry.
Nadja, thank you so much for this interesting description of the precision medicine omics work that's done at RI. Now, I want to change a little bit gears and come to other aspects of personalized medicine, more specifically pervasive sensing, and later then to mathematical modeling and what this can do to our patients. So, I mean, when you think about it, we see our dialysis patients 3 times a week for a few hours, say for 4 hours. This makes this 12 hours. It's about 7% of the entire week. We are totally ignorant about what's happening in those other 92, 93% of the week. And pervasive sensing is one way to help us to understand what's going on actually in these time periods between dialysis sessions. And we are very fortunate because industry is developing many tools that actually support these pervasive sensing aspects. There are wearable tools, ingestible, implantable tools, or even now sensors that are built into the environment. So the important thing is we want to understand better patients' health conditions outside of the dialysis. And to that end, we started a large trial. It's called Prospective RRI Interdialytic Activity Monitoring Exploration, or PRIME study. It's a 1-year observational study where we enrolled 125 patients and we collected 95 patient years of Fitbit data. Fitbit, that's a wearable device that patients were asked to wear in essence 24/7/365. And we would collect data on heart rate, on steps taken, on sleep indicators. And to our knowledge, this is, to the best of our knowledge, this is actually the largest pervasive sensing database in hemodialysis patients. Now, while many analyses are underway, I just want to share with you some very minor but still important aspect here. So what Maggie Han did, she analyzed pervasive sensing data during the COVID-19 pandemic in 45 patients that still happened to be on the prime study when the pandemic hit New York City. It's actually really interesting. On the graph on the right, you see the weeks whereby week 0 indicates the March 22nd, so when the New York State lockdown started. And then you see the average number of steps in confidence interval. And what you can see, there is this dramatic decline in patients' physical activities. Now, something very interesting happened. And I want to draw your attention to the bottom left of the slide. 6 out of those 45 patients got actually COVID-19, and 5 out of those patients took steps that were way above the 95% confidence limit, the upper limit of non-infected patients. So we think they may have had a higher chance to be infected with COVID because they were just moving so much around. So this is, for example, one insight, but many, many, many more analyses are about to follow, and we collaborate with a number of groups. Now, just to change gears, something very different. At RI, in collaboration with the data modeling and simulation team with GRD in Germany, but also with academia, we developed a very cool concept, namely the concept of virtual clinics, of virtual patients, and of virtual clinical trials. What do I mean with that? So in essence, you develop physiology-based mathematical models, say a model of erythropoiesis. Then you create patient avatars or mathematical twins of patients by using patient-specific data to identify certain model parameters. And so in other words, you have in your computer, so to speak, a mathematical twin of a patient. And then you run trials on those mathematical twins and see how patients would react towards certain interventions. And we are currently running a randomized controlled trial of personalized anemia therapy, and Doris Fürtinger, Sabrina Kasper, David Jürgen, Alhaji Sharif are working on this, the first 3 out of GRD in Germany. And what's done there is really to create avatars of patients that are randomized into the intervention group. And then the model is adapted, and then it would predict the patient's hemoglobin based on specific interventions. And the intervention is, of course, the application of an ESA, of an erythropoiesis stimulating drug, in our case, Mircera. And the interim results actually look quite encouraging. And as you can see on the top middle panel, this is just the hemoglobin data from one patient that was quite cycling and oscillating, but then when the precision therapy would kick in, then the hemoglobin would stabilize. And the same is shown for a larger number of patients on the bottom center, where you see the hemoglobin values. They were quite oscillating and cycling before the intervention, and the variability would go down substantially. Now, it takes, of course, a village to pull off a randomized controlled trial, and some of the key members are shown on the bottom left. I won't go through all of them, but it's a diverse team of mathematicians, of physicians, of data analysts, of clinical research coordinators. So it's a wonderful project, and we hope to get first results by the end of this year. Another aspect of personalized medicine is adaptive ultrafiltration control. As you know, during dialysis, not only solutes are removed, but also fluid is removed. And the question is actually, at what rate should fluid be removed? And we studied a few years ago and published this with Priscilla Preciado as first author, relative blood volume ranges that are associated with improved patient survival. So when fluid is removed, hematocrit rises and the relative blood volume declines. And what we asked ourselves, are there actually specific relative blood volume ranges throughout the treatment that are associated with better survival? And we were able to identify those ranges. They are shown in the figure and also in the little table on the right side. Interestingly enough, 2/3 of the patients were in an area where there was just an insufficient decline of relative blood volume. And only 1/3 were within those favorable ranges. Very few patients were actually below those ranges. Now, this information can now be used to inform an automated UF feedback control. And so the way how this works, that relative blood volume is measured, and the PI, proportional integral controller, tries now, together with a few other constraints, to change ultrafiltration rate in a way so that the patient's relative blood volume trajectory goes through these favorable ranges. And we studied this actually in a number of patients with a slightly different approach, where a healthcare professional still had to sign off on the ultrafiltration rate recommendations. But as you can see on the right side in the graph, with standard of care, about 1/3 of patients were in favorable relative blood volume ranges. With adaptive control, this more than doubled. Over 70% were brought into those favorable ranges. And so we think that there is many benefits to UF control, and they are all shown on the right side. So we are currently working with the FDA to get this to get an approval for that device. Now, something again very different, but another aspect of personalized medicine is will be shown in the next example. So what patients with AV fistulas and graft experience on occasion is the development of aneurysms. These are widenings of the vessel. And apart from not being cosmetically really attractive, they are at risk for rupture. And when an aneurysm ruptures, many patients with ruptured aneurysms actually die. They just bleed to death. And so we're thinking about the solution, and we built on the pervasive availability of smartphones. And so we would develop under the leadership of Hanji Zhang. She's an expert in biostatistics and AI. And we would develop a system that analyzes pictures from aneurysms using what's called a convolutional neural network. Without going too much into the details here, so this convolutional neural network that resides in the cloud can analyze these images and then identify aneurysms as advanced or not advanced. Now, we collected a large number of images, and those were actually adjudicated by a team of vascular access experts under the leadership of Dr. Murad Zor from Asura Health. And then the neural network would learn based on these images and the adjudicated stages what new images would look like in terms of aneurysm stage. And we developed this app. So what's done is that one takes a picture, the picture is of the vascular excess is automatically sent to the cloud. Then within a split of a second, the probability for certain aneurysm stages are calculated and they are sent back to the user. So we studied over 1,200 images, and they were classified as shown here. And you see on the left side, not advanced aneurysm. On the right side, you see very advanced aneurysms. Some of those actually at the dangerously advanced stage, and they would require immediate intervention by a vascular surgeon. So this brings me back actually to the starting point. I mean, we live in a very diverse world, right? We are, I mean, it's our patients are diverse, and with respect to background, with respect to gender, with respect to ethical background, with respect to social indicators of health. I mean, it's just a very, very diverse world, and our goal is really to respect this diversity and to define treatment options that are optimal for a given patient, not for the entire population, because it's we are treating patients and not not entire populations in this sense. Eventually, when the treatment of specific patients is getting better and better, of course, as a result, the care for the entire population will become better. No doubt about that. But the goal is really to improve the care for individual patients. And because we have such a diverse world, we strongly believe that we also need a very diverse team to work on that. And this is just a part of the RI team. And it makes me very proud to see that the team is as diverse as the patients we serve. It's a group of people with different backgrounds, skill sets, from physicians to mathematicians to biochemists to statisticians, epidemiologists, clinical research coordinator, and so on and so forth. And I think this is necessary to really address the key problems our patients face. So I want to thank you also in the name of Nadja for your attention, and we are looking forward to the discussion.
Thank you, Peter. Great, interesting aspects in both talks, and give my regards to Nadja Grobe. And let's welcome now Shaolin Wang. She will step in the discussion today and will discuss with us the questions you have given into the chat. You still have the opportunity to ask your questions and introduce them in the chat box on the right bottom corner. So I will ask the studio to collect and order the questions, and we'll bring up the first one, which we can discuss. And this goes actually to both of you. And I will read the question is, believing that for developing reliable precision medicine, we need a large number of patients. How far are we from a precise applicable biobank for clinical practice? So who will take the question? To do the research out of a biobank.
Yeah, I'm more than happy to start with this. So actually, I mean, this is true. I mean, in order to really understand the biological variability, and also in order to be able to get insights into the variants, we need a large biobank. So there is internationally, there is, of course, major efforts underway to also funded by the NIH, for example, and other funding institutions to create large biobanks. We have beat on genomic data banks, and of course, FMC is playing also an important role here. I mean, we have started developing biobanks, I guess, something like 2 years ago. So it's standard in all our clinical research that we ask patients to consent to having their specimen biobank, then most patients agree on that. Now the question is, of course, and I think that's the core, where do we stand with that specifically in, I guess, in the dialysis world? I think actually that we are on a good way to understand using biobanks, for example, specific aspects of peritoneal dialysis treatment. So I would really think that in the not too distant future, we may get insights using metabolomics research into transport status, and this then may translate into clinical applications. I don't know, Charlene, you have more profound understanding of the biobanking efforts. Maybe you want to add to that?
Yeah, I think you said everything correct. We are working on the biobank, and we are trying to determine how much volume we want to biobank for each patient at each time period. So in the future, we can implement it to all our patients.
So let me connect my question to this important one, is that I have learned the training set and then the validation set. And I think you are doing a prospective study using this mathematical modeling and machine learning. So my question to you, maybe Dr. Wang, is how big must the cohorts be, so the biobank sampling, be in the training and then in the validation cohort? Is with this machine learning, is this still an example of 1,000 patients, as the question here is large number of patients, or do you go along with only a few hundreds with this new technology? That's my personal interest.
Yeah, I think for at the current stage, we are recruiting 1,000 patients to do this machine learning. After we get a good pattern, we can use it to patients. But you think 1,000 patients is a lot, but if I would do everything automatically, it won't be too much work for us.
That's interesting. That's good. Yeah. Okay. 1,000 still is a lot. So yeah, okay.
But maybe I mean we have not yet the experience with the specific PD project, because I mean this is starting and ramping up. However, and what we have learned from the aneurysm project, we used some 1,200, 1,300 patients, and we were indeed able with 800 or 800 to 900 patients in the training set to achieve excellent areas under the ROC curves, excellent predictive value in the unrelated test set. So just to give you an idea, from this specific example in the aneurysm classification project.
So let's go to the next question, and I'm asking the studio, and it's coming, and I'm reading it as: current symptom-based approaches may potentially lead to misdiagnosis. Do you think the traditional nomenclature in kidney disease should change to something that's more precise?
Yeah, this is a very, very interesting question, I have to say, and a pretty difficult one. I mean, thinking back historically, I mean, kidney diseases were just classified as nephritic or nephrotic, for example. Now, with the advent of biopsy, the classification became more nuanced than with the development of different methods to evaluate biopsy, electron microscopy, histochemistry, etc. It became even more nuanced, and now with the advent of genetics, I mean, we are narrowing down specific entities, and I certainly would think, and new entities come up, of course, that were unbeknownst earlier. So I have no doubt that that the classification of kidney diseases will become more nuanced, that new kinds of kidney disease in a certain way will evolve, and that reclassifications of, for example, of tubulopathies, just as an example of minimal change, that those will become necessary as we learn more about the underlying mechanisms.
The beauty of the questions going to both of you is that Dr. Wang has some seconds and ideas to reflect. So do you agree with Peter, actually your boss, or do you have another idea?
I agree with Peter. And also I think both traditional way and also the new precise medicine should be used in the modern world.
Good, let's go to the next question. And this is going to you, Shaolin. Do molecular signature and fingerprints change with age? And does aging influence the analysis of biobank and biomarkers?
That's a great question. The short answer is that we don't know yet. That's why we need to recruit 1,000 patients at a different age and a different time of peritoneal dialysis, and we are going to try to classify it on all kind of a perspective and see if anything is to see if we can find any pattern or not.
Very good. Do we have another question? I also have two on my list, but let's do this one. There is a tension between EBM and individualized medicine. Can precision medicine close the gap, or isn't there a danger that evidence is necessary for many small patient groups?
Yeah, I mean, I'm not sure if I would call this a danger. I think that indeed, and we have seen this, say, with certain glomerular diseases, where a more precise definition of the underlying cause just requires a different treatment. And of course, there is evidence necessary to show that this different treatment has clinical benefit. So yes, I think when we have smaller groups of patients that are defined, say, by a specific genetic mark or by metabolomic fingerprint or something of that sort, that and when we plan the interventions, yes, of course, we need to generate evidence that this is the right approach. But I think that I would think that the medical community is quite willing and eager to provide that evidence.
Good. Then I will ask another personal question I came in mind, that in respect to pervasive sensing, I learned that the car industry is very interesting in getting signals from those who are driving. The car is getting the signals to react to certain sensing. Are there any links that is the car industry looking at you, or can you look into this, or are these two industries totally unrelated, or is it a different world?
Yeah, I would think that there is some intersections. I mean, for example, in the car industry, something that's pretty advanced is to detect drivers who are about to fall asleep. And so they detect subtle signs on on eye movements, on the movement of the steering wheel that are indicating of patients possibly falling asleep. And I think that's, we could learn at least from methodologically, what are they doing? What kind of, say, AI systems are they using? Which ones are particularly effective there? I mean, the interesting question is if, for example, some signals that say from drivers that are captured by cars have any medical ramification. To be honest, I've not thought about this, and so I don't want to speculate too much, but it's certainly an interesting thought to spend a little bit more time about that. So I think the overlap is mainly when it comes to methodology.
Yeah, I agree with Peter. I think this is definitely a very interesting idea. And the AI, like between two industries, definitely there's something to share. See how they make it work, and we can just get some knowledge from them and to make our work as well. Like even the Fitbit, the idea is similar to the car sensing, I would say, but we are just sensing the heartbeat or other things.
Thank you, Dr. Wang. So we take another question from our pool, and to Dr. Professor Kotanko. Can mathematical modeling combine different pathologic complexity subtypes into one subtype? Or are there already applicable clinical trials on multiple comorbidity?
So, I mean, the way how this mathematical modeling that we, for example, use for bone metabolism, for anemia, for acid-base metabolism, and other use cases is that we describe in a very generic way the physiological or pathophysiological process. And then, of course, once this generic model is created, then one can tweak or certain aspects, like, for example, with anemia, one can tweak red blood cell lifespan because the patient has a tendency towards hemolysis, or one can tweak the responsiveness of the bone marrow, just to name a few examples. And then, of course, these various components can be, if necessary, can be combined. So yes, in that sense, different pathologies can be combined in a model. Now, what's much more complicated is, for example, what if the patient in who you want to understand anemia has some comorbidities such as cardiovascular disease and underlying inflammation? How does this inflammation then impact anemia in this patient? This would require extensions of the model with other pathways. I think it's possible in theory, and actually with respect to hepcidin inflammation, where we're working on that. But this is such a holistic model, all-encompassing. These ones don't exist to the best of my knowledge. I mean, Guyton tried this with blood pressure, but I think there were some problems with that. But currently the models are narrow to and really targeting very specific organ systems, physiological pathways. I'm not aware of really an all-encompassing physiology-based model.
Okay. I have another question for very hypothetical, speculative. I was carefully listening to your talk, Peter, and I don't know whether I captured the number of plasma volume contraction in response to ultrafiltration rate. I don't know, but I'm asking my question that there is a drug on the market, the SGLT2 inhibitor, which are doing osmotic diuresis, natriuresis, and they always lead to a plasma volume contraction of 7%. And this has been repeated in several studies. And I always was wondering why 7% plasma volume concentration? And why not 8%? Why not 6%? Is this something the body, the natural body is reacting as an optimal thing? Do you see a correlate in ultrafiltration?
No, I don't see a correlate in the ultrafiltration in dialysis patients. However, what you're saying, this is really fascinating, and I would love to learn more about this. I mean, I could immediately think about several physiological mechanisms that kind of create an upper bound of volume contraction. For example, when it comes to oncotic pressures, when it comes to the activation of the renin-angiotensin system, of ADH system and vasopressin and so on. So it would be actually really interesting to look into these components, because then what does the, what does the interstitial fluid dynamics look like with this volume contraction? Is the plasma refill rate, does this reach a specific limit? This would be very interesting. And I mean, we have developed models of capillary fluid dynamics, and there is other groups working on this. It would be really interesting to look into, to what extent such models could explain actually that apparently there is a 7% threshold or a 7% limit that's eventually reached. Yeah, I would love to continue a conversation about that.
In order not to get arterial underfilling and myocardial stunning and what we all have learned from papers. Good. Xiaoling, any final comment from your side in respect to all questions? Is everything nicely answered, or any emotion from your side?
And we get some great questions today. Yeah, make us need to think more about the precision medicine and trying to make our work better.
Good.
Thank you.
That is a good final comment. So I thank you for these lectures and useful and productive discussion. And we are coming to the end of your interaction and presentation. Thank you again. Thank you. So now we are going towards webinar number 18. On the next occasion, which is September 1st, we hear from Dr. Franklin Maddox and Stefano Stewart, perspectives from a large dialysis provider, lessons learned from the 18 months after the COVID-19 pandemic. And certainly a very exciting 2 lectures, September 1st. Please mark your calendar. And then finally also, we are walking towards our Live 2021 Nephrology Congress, which will take place virtually and in part on-site, and from September 15th to 18th. And I have seen beautiful elements, some pre-registered talks already, and some live components. And you will really be excited that this Congress will set a new standard of technology into the digital world, but also combined with our previous old face-to-face components. So please save the date. The registration is already open. We have already an exciting group which registered, but we can always digest more in this digital or virtual world. And I can guarantee this will be exciting. So thank you for today, and looking forward to September 1st and the Congress September 15 to 18. Thank you.